Okay, there we go. Back to the tip of the iceberg. So that's taken me 10 minutes, so I'll have to speed up. So this is a kind of a basically some of these topics have come up, the relationship between environment, genes, and behavior. And so where is my talk going to focus on? It's going to focus on this part, intracellular signaling, which is sort of intermediate between proteins and the sort of local synaptic and membrane properties that Anto started talking about that obviously together collectively reorganize local circuits and larger neuronal populations to mediate interactions between genes and behavior and environment and genes. And so things like drugs of abuse, addictive drugs, all initially are perturbing intracellular signaling. And so the challenge is to try to understand that, as I alluded to earlier. So this is a timescale issue. So basically, in the initial, the fastest things that are happening in cells and ion channels are caused by rearrangements and protein conformations are occurring in the millisecond range. The things that we're going to start talking about, protein phosphorylation and modulation, are occurring within the sort of second range to many seconds. And then one starts to see transitions into changes in synaptic plasticity that are dependent on protein trafficking initially over a period of tens of minutes to 90 minutes or so. And then you start to transition into new transcription, new protein synthesis, which causes long-term changes and transitions into protein synthesis-dependent plasticities that ultimately affect neuronal growth, synapse formation, cell survival, other morphological alterations that really are the things that are starting to last for days, hours, days, weeks, months, years, that ultimately in an addicted animal are the consequences of things that started back here when we activated a dopamine receptor for a few hundred milliseconds. Okay, so that's really one of the challenges, as I alluded to earlier. So we're going to talk about dopamine, our favorite neurotransmitter. There are about 400 to 600,000 dopamine neurons emanating in the midbrain, projecting to the striatum, to the cortex, particularly the prefrontal cortex. In mice, there's about 20 to 30,000, or somewhat more in rats, obviously. And so what we're going to be focusing largely for this talk is on the input to the striatum for a number of different reasons. Obviously, we've heard a lot about various parts of the brain and how they interact. But we're really going to focus on these midbrain dopamine neurons projecting to the dorsal and ventral striatum. And in fact, for the sake of most of the biochemical studies, the work has been done in the dorsal striatum mainly because of accessibility. Although many of the signaling pathways that are seen in the dorsal striatum are found in large part in the ventral striatum as well. So this just sort of illustrates schematically in a slightly different picture of the main pathways that we're talking about. So the neurons, the dopamine neurons that emanate from the nigra or the VTA, shown in blue here, they're neither positive or negative. They're really modulators. They can go in various directions depending on their actions. And they synapse onto the major output neurons in the striatum, the medium spiny neurons that make up about 90 to 95% of all of the output neurons from the striatum. What they do there is mainly serve to modulate glutamatergic input from cortex that then is relayed through GABAergic synapses, either in the so-called direct pathway or the indirect pathway back to the nigra, and then from then on in a circle back through the thalamus back to the cortex. And this is occurring topographically, different parts of cortex synapse to different parts of striatum, different parts of nigra, thalamus, and back to cortex again. So this is sort of a fairly classical view. There are details that remain to be elucidated, but I'm going to use this sort of fairly simple classical view of the circuitry to discuss what's happening locally at these synapses when dopamine influences glutamate. And this is shown again somewhat schematically in a little more detail. If you take, in this case, we're looking at VTA projections, but basically to the nucleus accumbens, the idea is that the dopamine neurons, although there are not many of them, they have the ability to form many hundreds of synapses locally on the dendritic spines of these medium spiny neurons. And this is a picture of a typical medium spiny neuron with its dendrites and many hundreds, if not thousands, of protuberances, which are the dendritic spines. if you look at these in a little bit more detail, what you see is that the glutamate, synapses are releasing glutamate onto the sort of the top of the spine here, as shown at the PSD, or the postsynaptic density area where the glutamate receptors are. And based on immunofluorescence data, the dopamine receptors, most of them are at the sides of these spines in a place that you can see that they would have the ability to modulate the local glutamate signals. And to cover this morning, this is a drug abuse lecture series, but we like dopamine because dopamine does a lot of stuff, and Anto went through these already, and we have projects that relate to schizophrenia and also to Parkinson's disease ongoing in the lab. So how do dopamine receptors work? Okay, so 5 dopamine receptors. They're subdivided into 2 classes, 2 general classes. There's the D1 class, which is the D1 and the D5 receptor. And these, as we started talking about, these are GPCRs that couple through this heterotrimeric G protein, which consists of an alpha subunit and the beta-gamma subunits, these accessory subunits. And these are localized close to the receptors, but upon ligand binding, there's a change in the structure of the receptor that allows these to be recruited and to be locally activated, such that the alpha S, or the G-alpha off in this case, which is normally in the GDP form, is exchanges for GTP, and it's the GTP form of the alpha S that's able to then activate adenyl cyclase, for example, and generate cyclic AMP, which in turn will start to activate things like protein kinase A and cause signal transduction. Now, in contrast, there's the D2 class, which kind of do the opposite. And so they have the ability to work through inhibitory alpha subunits to suppress the ability of adenyl cyclase to stimulate cyclic AMP. So there's a mutually exclusive antagonism between these 2 pathways. In addition, sorry, I'm getting ahead of my time, and I'm not going to get into a lot of discussion of all the things that dopamine receptors do. They've all been knocked out. There are various forms of them. They've been manipulated up and down in combination, and largely, I'd say, if you go back even to mid 2000s, there are a number of reviews that you can read that will kind of explain pretty clearly that we really understand at this level how the different dopamine receptors are playing roles in mediating the effect of dopamine in different neurons. And as expected, they play critical roles if you knock them out and control basic motor function, cognition, reward, various other behaviors that you expect that are involved in these parts of the brain. So interestingly, when actually I was preparing this lecture to go to Barcelona last year, and at the time, this paper came out in August 1972 from Paul Greengard and John Kababian, and PNES decided to do a retrospective on this paper. And so Saul Snyder wrote a retrospective, and he called up a number of people to say, "What do you think of this paper?" You know, so this was really the start of the idea that neurotransmitters like dopamine could modulate signal transduction pathways. And so I actually went back and read it. I hadn't read it probably in 20-something years. So this is Paul Greengard. He's a little bit older now, but he's still working in lab. John Kababian I think is pre-retired Paul. Anyway, I won't read through the details of the abstract, but a few things caught my eye. One is basically they made lysates of caudate putamen. They stimulated with dopamine or norepinephrine or isoproterenol, and you see this very nice generation of cyclic AMP with a half maximal level of about I think they say 4 micromolar. And then they do this nice experiment, which actually was really an important part of this study, where they do the same experiment. They stimulate with dopamine or without dopamine, and they do it with a dose response of haloperidol. So what's haloperidol? For the low levels for D2, right? So if you put their data together, they concluded that that here it says the stimulating effect of dopamine is blocked by low concentrations of either haloperidol or chlorpromazine, agents known to block the actions of dopamine in the mammalian brain. But from what I just told you about the way the receptors work, what's wrong about these?
Shouldn't be through cyclic AMP.
In fact, it should go the opposite way, right? So if you go back to this, you should actually, haloperidol should actually increase cyclic AMP because it should block this pathway and potentiate, and that's actually what we now know is the case. But in fact, in this isolated cell system, they got the opposite result. It didn't change history, but it's sort of interesting how the origins of these were sort of limited by the methods that they had and the availability of knowledge that they had at that time, because they didn't know there were D1 and D2 receptors. They just,
why does it block it? Is it the concentration?
because it's really high concentration, I think. I think it gets into the issue that crosses over and generally blocks the D1 receptor.
Is that micromolar?
Yeah, this is micromolar. This is up to almost 100.
So for haloperidol effects specifically on D2, is it?
It's much lower. I don't remember exactly the IC50, but it's much lower than that. So, so it was good that it didn't change history, but you know, you have to think about these things when you get data, which is interesting. Yeah.
I think there's another layer of complexity to it because the D1 and D2 receptors are not in the same cell for the most part.
I'm going to get to that. That's going to be a major bit.
Okay. I was thinking you might need something to activate a different GS pathway to see the
Exactly.
effect.
Yeah. Anyway, I'm going to get to that, and we'll come back and discuss. But those are important. That's an important point that I'm going to address in a couple of different ways. So one other thing that the dopamine receptors do, D2 class, is actually they can take this beta gamma component and actually modulate directly a number of different ion channels. Okay, and this actually was a hugely controversial area of research for a while, but we now know that this is really true. So this is kind of the model for that. So basically, dopamine receptors can interact with D2-like receptors, cause release of the GI in the GTP form that can do its thing, but then release into the membrane still the G beta gamma that can be recruited to potassium channels, the GERT channels, but also calcium and sodium channels. And I'm going to show you a picture of one of these, a real picture of how this occurs that just came out in Nature from the McKinnon lab. So, but things are complicated. You mentioned which cells the receptors are in. There are other things. This is from Anto and Mark van Zastra's a paper that came out last year, I think. And this, I won't get into the details, but what they found was that if they use things that perturb endocytosis of the receptors, so you all, particularly in opiate receptors, but in all the GPCRs, they go through a desensitization step that involves them being internalized into the cell. In some cases, they can then start to do things that they weren't doing on the surface of the cell. And this study suggested, in fact, looking at cyclic AMP generation using a fluorescent-based assay, that a significant amount of the cyclic AMP that was generated actually occurred once the receptor was going into the cell, if I understand the conclusions correctly. So there are a bunch of things like this that makes things more complicated. We touched on a few seconds ago receptor localization, but in fact, there's a huge amount of work has been done on receptor dimerization. I'm not going to go through all these various things. This is a review from Susan George. It just came out a couple of weeks ago. But you can see D1, D2 interactions, D2, D4, adenosine receptors interacting with dopamine receptors, NMDA receptors interacting with dopamine receptors interacting with serotonin receptors. Huge number of biochemical data that suggests that these things occur. They are real. But then the question is, are they in the same cells? You know, what are they doing? We don't really understand it. We don't have the reagents that are really available to specifically activate or deactivate a complex of a dimer, you know, a heterodimer compared to a normal single type of a receptor. So these are things which I think the significance is still remains to be elucidated.
There's a lot of controversy about the D1, D2.
There's almost every paper there's controversy, and
coming out soon that's going to refute a lot of that.
you know, the same thing occurs in the opiate receptor world. There's a hugely controversial.
So Angus, do you want to, sorry, do you want to spend a second saying what Devon just said? This D1, D2 method has been going on for 20 years.
I can, no, I probably, because I'm a little bit biased, I would probably say the wrong thing.
Controversy is?
Well, I think the controversy is, I think my understanding is that some of the data is based on biochemical data that says the receptors can actually interact with each other in some cases. In other cases, it's based on fluorescent-based assays that say they can be close to each other in the same vicinity. Most of that data, I'd say, is okay, but it's limited by the methodology. But the functional data is to try to find something that will be a specific agonist, for example, for a complex versus a non-complex. Now, that's, I think, when it starts to get iffy. Are there really functional effects? And we're going to get, maybe I'll get ahead of myself here. I'm going to come to the conclusion based on other data that suggests that most of the D1 receptors are in one cell type and most of the D2 receptors in the other cell type. So if you do have a few cells or a few molecules, you know, how physiologically relevant are they compared to these major signals that are occurring? So I think that's the issue is within the big signal, you know, are these little signals really important or not? And I think that's...
So I just want to make a comment as a synaptic physiologist. There was a paper many years ago in the Lupus Academy where a guy showed, Satoshi Kimoto, that rats will only self-administer a combination of D1 and D2 agonists together into the accounts. They don't self-administer a D1 agonist or a D2 agonist. We started from that and we did electrophysiology in the accountant's shell, same place. And when we but applied dopamine or a D1 or a D2 agonist, we saw exactly the same thing. We couldn't evoke firing of medium spiny neurons, which are the 95% of the cell, unless we used a combination of a D1 and a D2 agonist. But, as Angus was saying, we were never able to prove where the two receptors were. We only know that we need both of them to be activated at the same time, but where they sit,
Yeah.
it's, and how they interact with each other.
So I mean, one
Could be circuitry.
so there could be, you know, we know there's a lot of recurrent collaterals coming back from the medium spinies back to themselves, and they may be coming from other places. We really don't have a handle on that yet, and so I think that's, you know, I think one of the reasons that we need to not we in addition to some of the methods, optogenetics, and we need biochemical methods that will allow us to actually go in and check within the same cells that we are doing these other experiments. Okay, so I'll skip through this part really fast. There's just pictures basically, but we know now a lot about the structures of these things, how they work. This is an early structure of rhodopsin and complex with the active form of the G-alpha protein here, the G-beta gamma. But we, you know, almost in the last 2 or 3 years, almost every week or month, there's a new structure. We know this is an example of a D3 receptor in a complex with a D2, D3 selective antagonist. So this sort of high-level resolution is actually going to help us get better agonists and antagonists that will be useful in these sort of questions that we're raising. Structure of A2A receptors bound to antagonists. A bunch of opioid receptor structures came out last year. There are more coming out steadily. We know exactly how adenyl cyclase is activated. This is a picture of the part of the active site of the adenyl cyclase in a complex with the GTP-bound form of the G-alpha S, showing this nice structure which affects this region called the switch 2 region that then allows us to interact and activate the channel. I mean, the cyclase in this case. This is a recent paper from Rod McKinnon's lab. This is the same sort of thing showing the activation of GERK channels by the beta-gamma complex, and we now have these very high-resolution structures showing exactly how the beta-gamma is able to interact with and perturb the pore of this, and also in this nice paper how lipids like PIP2 play a role in modulating the activity of the channel. So this stuff is coming out all the time. There's a lot of detail, obviously, but I think it allows us to get a much more better molecular understanding of what's going on, and also to be able to design better drugs that pharmacologically perturb these systems. Okay, so that's kind of the first part. We'll switch back onto the next part, which is downstream. So phosphorylation and dephosphorylation. So we already talked about this a little bit. There are about 500 and so protein kinases in the human genome. About 428 of them are so-called serine-threonine kinases. The other ones are tyrosine kinases. These are different. They're a little bit different, but most kinases, almost all kinases, look sort of like another kinase. In contrast, if you look at serine-threonine phosphatases, there are only about 20, which is a big, an order of magnitude of more difference. And so one of the things that our lab has been working on over the last 20 years ago is to try to understand the mechanisms that modulate these proteins. And I'll touch on some of that data in a few seconds. I've already said a lot of proteins are phosphorylated. Maybe it could be 100%, but certainly 60 to 70% is sort of the number that people put around. So, if you were working on some system, and most of you are, I think, not molecular biologists, but if you bumped into a protein of interest and somebody said, well, is it regulated by phosphorylation? What might be the first thing that you would want to know? Any ideas? Any thoughts? Maybe
Consensus sites.
So what are consensus sites? So you might want to ask which kinase phosphorylates it, right? And so I'll touch on that. And that's a question of how does the kinase, these 400, 500 kinases, how do they discriminate between different parts of the molecule and pick one phosphorylation site to change? What might you do if you just wanted to know where the phosphorylation sites are? Because you wanted to maybe mutate one that might be important in synaptic plasticity, like serine 845 of the AMPA receptor. What would you do?
I think there are two approaches. So if you have the access, you could do like a proteomic, or you could basically just look for the phosphorylation
by mass spec or something like that, right?
Alternatively, you start making deletions in your
Right.
construct.
Those are exactly what you would do, but or would have done. But in fact, these days, almost all the phosphorylation sites are known by huge mass spec data. So there are databases with hundreds of thousands of phosphorylation sites. So before doing anything, you turn your computer on and you put your sequence in, or your protein, or your name, or your accession number, and it will come out with all. It takes a few minutes to learn to go through it, but that would be the first thing you would do these days, Jonathan.
So a lot of these signals are similar or the same, and in a cell, is there some way to segregate that the cell actually segregates the signal?
I'll touch on some of that in a second or two. The answer is definitely yes, but it's to resolve with the resolution that you'd want. You not only need high resolution imaging and reporter probes. You need to look at combinations of them, and that becomes more challenging. But localization, it's like real estate. Localization is really critical. So here's the human kinome. We're going to just really very, very rapidly talk about a couple of kinases. The ERK MAP kinase and PKA I've already introduced for various reasons. PKA regulates plasticity through phosphorylation of GLR1 and transcription factors like CREB. ERK is important in mediating the transduction of signals from the surface of the cell into the nucleus. There are other kinases that were mentioned briefly. CAM kinase 2 is involved in LTP. CDK5, something we work on. CK1, Jonathan mentioned yesterday, is being important in psychostimulant action. And all of these kinases actually phosphorylate our favorite protein. So PKA is a classic kinase, one of the first ones that was discovered. It's regulated by what's called an autoinhibitory pseudosubstrate mechanism. So for people who remember what Pac-Man were, the catalytic subunit up here is the Pac-Man, and the R subunit is something which is trying to eat them from the inside. So basically, it would put a piece of the protein, which is shown here in this red inhibitory segment, into the active site and mimic the normal peptide that would be phosphorylated. When cyclic AMP comes along, that would bind 4 of them to the regulatory subunits. This would then displace these away, and the enzyme would then be able to go and find its favorite substrate. The ERK pathway is a little bit looked at somewhat differently because it was something that was discovered when people were trying to understand how receptor tyrosine kinases were acting on, particularly on nuclear events. So any receptor tyrosine kinase has been mentioned this week that are important for what you guys study? Track B, BDNF, exactly. So BDNF would use this pathway, this canonical MAP kinase pathway, where it activates locally small GTPase proteins like RAS, activates a kinase called RAF, that then activates another kinase called MEK, which then activates ERK1, and then ERK goes into the nucleus and regulates a bunch of transcription events. And these kinases are part of what's called a kinase cascade, and they work by virtue of being able to phosphorylate residues within. This is a kinase here. For ERK, this is the active site is in here. The ATP would be sitting in here. The substrate would normally sit in this sort of position. And there are a couple of phosphorylation sites that have to be phosphorylated by the upstream kinase. They get phosphorylated, the structure changes, and the enzyme becomes active and allows the target peptide to sit here. Okay. So phosphatases come in a little bit different flavors. There are actually more types of phosphatases. Kinases tend to be are really all the same gene family. They work the same way, but phosphatases are more complicated. They split into two large groups: the serine threonine phosphatases and the tyrosine phosphatases. These are structurally, enzymatically quite distinct. There are a number of interesting guys. I won't talk about atypical enzymes, which are becoming more important. Within the serine threonine phosphatases, the type 1, 2A, and type 2B separate from the type 2C. Type 2B is called calcineurin. I don't know if Marina mentioned calcineurin in the context of nicotine action, but it certainly plays an important role. Calcineurin is a calcium calmodulin-dependent phosphatase that also plays a key role in long-term depression. It's part of the switch-off mechanism. There are a bunch of other guys. I won't get, for the sake of time, get into details, but these can be further subdivided into various subclasses. But the key difference is that these different subgroups of phosphatases are quite mechanistically distinct and are regulated in different ways compared to protein kinases, who kind of tend to work by the same way. Okay. Let me just skip over this for the sake of time. So one of the important things that I want to get out of this part of the talk is why do we have protein kinases and protein phosphatases? Are they, is one the major regulator, or are they both working together in concert, right? And so there's sort of been a dogma in the field that the kinase is the thing that's the turn-on switch. It's where all the regulation is, and the phosphatases basically are generically dephosphorylating things. But the reality of that is is not the case. And I'm just going to go through a few examples of what makes it very clear that that's not the case. So in fact, phosphatases are not simple off switches. So if you look at the SARC family kinases that are important in development and cancer, or if you look at cell cycle progression, in fact, the key step is actually the action of a phosphatase to turn on a kinase. So the phosphatase is actually upstream of the kinase. So they're not off switches, they're actually on switches. They obviously have an important role in setting the baseline. So if you have, you look at the steady state phosphorylation of a phosphorylated protein and you stimulate it with some sort of agonist, it will go from a low state up to a high state. But where you start here, if there's a lot of phosphatase activity, then this is going to be low. If there's not so much phosphatase activity, you could be up here. So the balance between the phosphatase action and the kinase action is really the critical thing. In fact, most of them, as you might think, based on undergraduate lectures on this sort of biochemical topics, these things are kept in balance. The balance is really useful because if you're at balance, you can move one way or the other and get much more control. And so the critical thing is this balance and how exactly the baseline state is set, because that can lead to the ability to amplify signals, okay? And I'm going to talk about a couple of examples, but you can imagine that if you have a strong phosphatase activity and a weak kinase, that if you increase the kinase in the face of a strong phosphatase, it's going to be harder to get your signal up. But if you turn off the phosphatase at the same time as you turn on the kinase, then you really have a lot of power. And so the Hill coefficient of your activation curve is going to be significantly greater. So amplification is key, and phosphatases are really important in that. You can go a little bit further if you want to take plasticity, for example, in a neuron. You can start to make these more fancy amplification mechanisms and build bistable switches and build in a lot of ultrasensitivity. And the way you do it typically is to take regulation of both the kinases and the phosphatase and add more layers on that give you more ability to shape that response. With a little bit more intuition, one can then start to design oscillators, and in fact the way that the circadian rhythm works is because of a balance between kinase and phosphatase activity working on the proteins and the related transcription factors that are regulated by phosphorylation, come out of the nucleus in a time-dependent fashion, are then dephosphorylated or phosphorylated and go back into it. So you can use these phosphatases and kinases in a time-dependent way to design various types of systems that you would like to control, and these are obviously important for neurons. So this gets to Jonathan's question in part. So one of the key things about phosphatases in particular is that they impose a huge amount of temporal and spatial specificity. The temporal part kind of is illustrated by these sort of things like switches and clocks, but in fact the phosphatases actually have the ability to be placed in discrete places in the cell, and they have a very fine control over the spatial specificity of any given signal. So you may get release of cyclic AMP throughout the cell, may activate PKA, but it gets turned on in a particular place, and then the phosphatases will turn it off specifically at that site. And so we actually see many cases complexes between kinases and phosphatases. You might think this would be kind of a futile cycle, but in fact these things are, you don't want to turn on signals for very long, and you only want to turn them on in the right place. And so the phosphatases play a sort of a hierarchically critical role in localizing and modulating the time of any signal in a particular place. Okay, so if you don't get anything else out of the presentation, one of the important things for phosphatases and kinases is that they're working together in a fine balance of very discrete places in the cell, controlling spatial and temporal signals. Okay, so we started to talk about that, and Chris already mentioned things, but how do kinases and phosphatases recognize the substrate, target substrate? So, so some of them work by recognition of, or kinases largely work by recognition of specific phosphorylation site sequences within a protein. And so if you look at PKA, for example, these are called so-called basophilic because they like arginines close to the serine or threonine. They like a hydrophobic residue. In contrast, the casein kinases are called acidophilic because they like acidic residues, aspartics and glutamate residues, and so on. CDK5 or MAP kinases like prolines after the phosphorylation site. They're called proline-directed. So that's probably the main way that these kinases select their substrate. So they scan through the sequence and they find one that they prefer, and then they will phosphorylate the serine or threonine. They also use, to some extent, docking interactions. These are analogous to what I mentioned for the phosphatases, but they will find regions of the target which bind to other parts of the protein that help to increase the specificity. They also interact with adapters and scaffolds that also allow kinases to be targeted at specific places in the cell, and that's a critical thing. But by and large, the kinases use these specific phosphorylation site sequences. And again, if you have your protein of interest, you can go to the web. There are things called scan site. You can enter your sequence. It'll give you a list of potential kinases that might phosphorylate sites within your protein. So the phosphatases, though, I think they work in a largely different way. And so I'm going to give one example from work on medium spiny neurons and phosphatases that we have done over the years. So here's a picture again of the medium spiny neuron. These are the dendrites and the spines. If you blow this up and look at a dendrite and a spine in cross-section here, here's the presynaptic terminal, mitochondria. You see the spine head, the PSD would be here, the neck, and then the dendrite. We're looking sort of down the dendrite along the microtubule motors that are in the dendrite. And what you see here is if you immunolocalize one of these serine phosphatases, an enzyme called PP1, it's highly localized in the neck and the spine, where you could imagine that it's perfectly poised to modulate signaling through glutamatergic signals, for example, or dopamine signals that are occurring within this region. Now, the phosphatase itself would not normally be there, and the reason it's there is because it binds to one of two proteins. One's called spinophyllin, and spinophyllin is an F-actin binding protein that is able to bind to the actin within the spine, and in turn it brings phosphatase 1 along for the ride. And the model, based on a lot of data I won't get into, is that this phosphatase is present in an active form, where it's able to locally dephosphorylate the important residues, for example, serine 845 and the glutamate receptor, and maintain it in an relatively inactive form. But if you stimulate, for example, with dopamine and D1 receptors, it activates pKa. It causes a phosphorylation event in spinophyllin that delocalizes it from this local site, removing the phosphatase away. So you inhibit the phosphatase, and then the kinase is then able to work, and you get this amplification mechanism. So this is one example of this sort of amplification mechanism that we see. And it's actually a little bit more interesting than that, because we did structural work with Wolfgang Petty's lab at Brown University. We've been working on phosphatase structure for a number of years. Now, this is one example. This is the catalytic subunit of protein phosphatase 1. It has two metals in its active site, a bunch of regions that you might imagine things sort of hanging onto. And so we formed, prior to working on the structure with spinophyllin, we had been playing with models about how substrates might be bound by phosphatases and be discriminated. And in fact, there isn't any similar discrimination between short sequences as you find in kinases. So in fact, when we started thinking, well, maybe phosphatases can bind substrates in different ways. So they may bind like this, they may bind like this, they may bind like this. The phosphate would still be in the active site, would still be dephosphorylated. And so when we got a crystal structure of the phosphatase in a complex with part of spinophyllin, this sort of runs around the back of the molecule, comes up and comes down this groove, comes down and has this helix, and then this PDZ domain at the front here. What we then realized, based on this structure and biochemical data, was that this structure actually provided selection. And so how does that work? Well, basically, we can't get any binding of this type of complex. We can't get binding of this type of complex, but we can actually still get this type of complex. And so based on this and a number of other structures that we have, we feel that the way the phosphatases are working is that they have these negative determinants that suppress a lot of substrates, but then select things. In this case, the biochemical data we showed was that the preferred substrate was the serine 845 or the glutamate receptor that was able. We don't have a structure of that, but that would be inferred by this data and also by some mutagenesis data that we did. Okay, so I think I'm almost at the end of 1 hour. Do we take a break or just keep on going?
Yeah, do you have two parts to your talk?
There's not really two parts. There's stopping points along the way. This is a
Okay,
stopping
I
point.
mean, we don't have to keep to this talk film. Don't worry about it. It's,
Why not go on for a little bit, and then there's another stopping point coming up fairly soon. Okay, so so that's kind of the didactic sort of stuff out of the way. That kind of basically introduces some of the players that I'm going to talk about, and the main part of the main part of the talk is really to talk about specific proteins that mediate the effects of dopamine and how they do it and how they involve phosphorylation and dephosphorylation regulation. And so this work has been going on for more than 30 years now, from the time that I joined Paul Gringott's lab at Yale, and then we moved to Rockefeller. And what we did at that time, we realized that at that time people had been basically chopping up the brain into the cortex and the cerebellum and spinal cord, and that was about the level of biochemical analysis. And so we realized this was very limited. And so Ivar Wallace and myself took up this project to actually more finely dissect. We looked at about 20 to 30 different brain regions that were defined anatomically or by neurotransmitter pathway, and started to look at signal transduction pathways that might be enriched in one or other of these parts of the brain. And really the first thing that popped out was the fact that there were a number of substrates, and this is summary data now, but a protein called DARP32, small protein of about 200 amino acids, another small protein of 88 amino acids we called ARP21, and another protein called ARP16, about 95 amino acids, all of which were highly enriched as we turned out, in the medium spiny neurons, shown here for DARP, in the sagittal section for the ARP21 we now call RCS, in this coronal section, and for ARP16. And so if you look at all of the enrichment of all the phosphoproteins through all the many different brain regions, this was the most striking, and really we started at that time to work on it because of that. We knew they're all phosphorylated by protein kinase A, threonine 34 for DARP, serine 55 for RCS, serine 88 for ARP16. And so what I'm going to do is really largely focus on DARP32, which has really been the prototype. Okay. So the thing that binds them together is that if we follow this canonical signaling pathway downstream of D1 receptors, we activate adenyl cyclase, generate cyclic AMP, stimulate PKA, and each of these three proteins is phosphorylated. And amazingly, what we found was that DARP32 is able to work by virtue of inhibiting one of the major phosphatase in the brain, serine phosphatase PP1. The RCS protein works in a somewhat different way. It sequesters calmodulin when phosphorylated, but it regulates the second major phosphatase, PP2B, and work which I'm not going to talk about, but recent work that we've been in. We now know that the ARP16 protein is able to regulate the third major phosphatase in serine phosphatase, PP2A. So I have to say, when we discovered this, that was interesting. When we thought, well, it's kind of a coincidence, but we now know that these three proteins all are modulated by dopamine, and their primary function is to regulate phosphatases. There must be something important about phosphatases in these neurons, and so that's what I'm going to tell you about for the next few minutes. So in terms of the way we think DARP works, it's sort of the same idea that I introduced before. We think it's basically working as an amplifier. And so the idea is this is the same pathway that I've shown already. Dopamine acts on D1 receptors. PKA is activated. It phosphorylates a bunch of substrates. They could be at the membrane. They could be in the nucleus. It could be in different parts of the cell. But many hundreds of proteins are phosphorylated by PKA. But at the same time, PKA phosphorylates DARP32 and turns it into a potent inhibitor of PP1. And PP1 is the phosphatase that dephosphorylates many of these substrates. And so by inhibiting the phosphatase at the same time as stimulating the kinase, you get a dramatic increase in the phosphorylation. And all of the data we have suggests that DARP is an amplifier, that if you manipulate it up or down, in a whole animal, for example, you shift the dose response curves for psychostimulants like cocaine one way or the other, consistent with this being something that's just setting the way the signal is amplified within the organism. And so, I don't know if I still have this connected up. Let's see if this will work. So, so this is kind of a summary slide, but DARP is really, for many people who have worked in the Gringard lab and in affiliation with the Gringard lab, we know that DARP is really probably the critical mediator of dopamine action. If you knock it out, particularly in mice, or manipulate its phosphorylation in mice, you significantly change the responsivity for anything that might influence dopamine, either psychostimulants, antipsychotic drugs, et cetera, et cetera. So DARP is present in all the medium spiny neurons, and it's really the key modulator. And it does so by virtue of the fact that PP1 is able to control initially local events of the membrane, but also more long-term events like transcription. And so this is, as I say, come out of the knockout mice work, and I'll show you a little bit later on, but there's also more recently human disease studies that link polymorphisms in DARP to various aspects of temporal and reinforcement learning into schizophrenia. And in no less an expert, neuroscience expert than Bob Dylan has acknowledged this in one of his radio talk shows a few years ago, and if this works, I will try to play this. Except I had it set up so that it would work automatically, but now I switched computers. I have to go back to... so you didn't know that when you publish your important papers that, in fact, Bob Dylan's guys are listening and analyzing it. Is it going to work? So I have it set up to work. Sorry about that, guys. Being challenged today. Oh well. Anyway, I'll try to get it to work during the break, and we can play it again. But basically, he very accurately quotes the yeah, but I don't have that on there. It's only on there, so that's why. I just turn it off. He very accurately describes the role of DARP, and particularly in its, he knows a little bit about optogenetics because he also talks about the connections between the prefrontal cortex and the striatum, but he says striatum in a very distinctive way, so I'll try to get it to work. Okay, so this, I actually modified the slide a little bit from discussions I had earlier with people here at the meeting, but it's, Jonathan has sort of alluded to, other people have alluded to, we study one phosphorylation event, and we have to study it in gory detail to understand it. But then we find that there are a lot more related phosphorylation events, and so then we start to say, well, how do we try to figure out how these things work together? And so I don't think there's any easy answer to that. You can certainly model signal transduction pathways once you get a reasonable amount of information about the component parts. But there are different ways to model. You can kind of model without data, or you can model with data. And if you want to model with data, you have to have good data. And so modeling with bad data is not a very good way to go. You can try to do sort of things like network analyses that don't necessarily need data about things, but just need to know who talks to who. I think because signal transduction is getting more and more complicated, the examples I'm going to show have 2 or 3, feedback or negative or positive feedback systems. My brain can still kind of handle that more or less, but once it goes beyond a few, it's really hard to figure out what's going on. So you have to kind of have some way to model, but I don't think we quite got to the level that we can systematically model in a way that we can feel confident that the models might be predictive of some predictive validity. So I think at the moment we have to kind of go back and forth, basically. We have to do a little bit of one, we have to go back and check the model and see if it works. And so the example of that is, I've told you about DARP, I've told you about one phosphorylation site, but it turns out that's only part of the story. So the pKa site is here, this is the one that regulates PP1. But in fact, there are at least 3 other sites that we well characterized. CDK5 regulates this threonine site. There is a negative feedback on this process. CK2 and CK1 have positive effects on this process. It turns out, of course, once you discover another phosphorylation site, then you have to figure out the phosphatases that regulate these dephosphorylation events, how they're regulated. It turns out, and I'll talk about one briefly, some of these phosphatases themselves are regulated by phosphorylation, and they have the kinases and phosphatases. So it doesn't take you very long to go from something you thought was relatively simple to a whole 20 or 30 things that might feed into the same pathway. And so how do you understand what's going on? If you knock one of these things out, how does it affect all the others? If I knock out a subunit for PP1, does that affect PP1's ability to do very local action, or does it have a knock-on effect because of compensation in other pathways? So those are really critical challenges, and when you read papers about somebody doing their favorite knockdown or knockout, you have to take some of it with a grain of salt because there are a lot of things that they don't know. So we actually have done some modeling. We've modeled with Paul Greengard's son, Leslie Greengard, is a mathematician. We spent some time modeling some parts of this pathway with him a few years ago. And interestingly, about 5 or 6 other groups around the world have modeled parts or all of some of the parts of the DARP signaling pathway. And although they've picked different ways to do it and different ways to, and different components, the good thing about it is they all agreed on the same thing, is that DARP's important. And to put in a simple sense, the terminology that he uses, it seems to add robustness to the system. In other words, if you don't have DARPs there, things don't work as well. They don't work as efficiently. A signal comes in, but the output is not as well coupled at the end point. And so if you put all these ROs and connections and components, what it does is it adds robustness. It makes the system, buffers high or low signals that you might want to filter out, and gives you a much more clear-cut connection from the input to the output. So I think that's probably going to be the case for a lot of signaling pathways, that they're going to add this sort of robustness and make things work like plasticity or whatever you want to do. Okay, so maybe we take a 5-minute break now, because this is a good breaking point.
Well, as I say, you've got flexibility. I mean, Nora doesn't start till 2.
But we have to have lunch.
Yeah, but 12:30 to 2 is the
Yeah, it's true. Okay.
So you have some.
I will try to get Bob Dylan set up, and then we'll go from there.
Sounds good.